Before that, I think a few words, I think, from FABA chapter side and invite Dhanraj.
Okay. Welcome all. Good evening and good morning. Happy Ganesh Chaturthi. We are here for another exciting webinar by Dr. Rao Prabhala, the topic being CAR T cell therapy, a magic bullet ammunition and immunotherapies. Know the pros and address the cons. And this is part of the FABA US chapter monthly webinar series we've been having since this past May. And this is the 5th one. And we are expecting it to be a very interesting session, and lots of registrants are there who will participate and join the call. So FABA US chapter, like the FABA India and many other branches of FABA across the globe, is interested in disseminating good science and practicing biotech at the highest ethical standard and integrity, and bring about global collaborations throughout worldwide as well as across the USA. And recently our update is that we finished a summer internship for high school students, and we graduated 46 high schoolers, and it was a very successful program. So moving on to the webinar series, Dr. Dhanraj Tiruchinapalli is one of our close associates and affiliates of FABA, and he contributes greatly to our membership board. So welcome, Dr. Dhanraj, and you may take over to do the welcome note and introduce Dr. Rao Prabhala, and let's begin the session. Thank you very much. Thank you all for coming here.
Thank you very much, Deepika, for that introduction. Appreciate it. Good morning, everyone. On behalf of the US chapter, it's an honor for me to introduce Rao Prabhala today. I looked at his profile. Dr. Rao Prabhala's career is a very interesting career span. He started off his career in 1977 from a small city in southern India, Guntur. As a plant biologist, evolved himself. As the evolution of the immune system, he evolved himself, moved on to do his PhD. He did his PhD in my current place where I am from. I'm from Arizona, so he did his PhD from Arizona. It's good to see an Arizonian. And went on to do his postdoc at Dartmouth and then become a faculty. And his life turned, and then he went transatlantic back to India. It looks like Mother India called him back to do some service over there. And then came back to the United States back and joined Harvard University and Dana-Farber as an instructor for 24 years. So his long legacy and career would be a good insight. And as Deepak did mention to us, the title of his talk today is going to be CAR T cell therapy. As you all know, CAR T cell therapy has started by Carl June and UPenn, and he is the father for this program. And it's good to hear from of these eminent speakers on CAR T cell therapy. And India has already started this program. The Indian government is very much interested in CAR T cell therapy, how we make a difference in the lives of the patients. And I welcome Dr. Rao Prabhala to this webinar series, and we look forward to speaking with him. Sir, please continue. Go ahead, and I'll hand over the podium to you.
Yeah, I'm going to stop sharing my slides, sir. Okay.
Thank you, Dr. Dhanrajan and Deepika and Redanagaru and all the FABA members in US chapter. And it's my pleasure to give a talk. So before I start, it's celebrating Vinayaka Chaviti. It's very educational, and it was the festival when I was growing up. 3-4 years, I make my own Matti Vinayakulu, make the clay slate, and have a little bit artistic in my nature. So I make my own. But now, when I took my children's back, it's becoming a little bit more commercial, Vinayaka Chaviti. But nonetheless, it's a good beginning on an auspicious day. So Today I will talk about CAR T cell therapy, which I have been involved in Farber since I came here, because I'm a trained immuno-oncologist, last 45, 46 years, with a little bit different flavors of infectious disease, nutrition, and medicinal plants, all sorts of things. But main focus is immuno-oncology. All my life in this country, since I came when I was 22, is cancer.
So Rao Garu, can you put on full screen?
Yeah,
And someone requests that the volume is low, sir.
Yeah, I think the volume is low, sir.
Yeah, yeah.
why? See, full screen, I will have a hard time to manage.
Oh, okay. So whatever that,
Right, that's the
yeah, yeah, you can go ahead with.
Yeah, I try to increase the volume on the computer. It says 80%, 85. I don't know whether this one has any control.
So you can
Can
raise
you hear me now? Can you hear me a little bit better or no?
better. You can raise your volume yourself
Yeah.
a little bit.
Yeah. So always, I'm a teacher. My grandfather is a teacher. My father is a high school headmaster and a science teacher. So I have a little bit teaching, and I teach with a passion, um, because my grandfather always says that you are useless to teach an intelligent student. Intelligent student doesn't need any teacher. Um, the teachers who are great will be the student who doesn't want to be in your class, who doesn't care why he is there. If you make him listen to you, then you are a great teacher. So you are looking for that one student. So we worry about in life a lot of stuff, especially even in this country. But the only thing we have in our hands or in our control is hard work. So I trust my hands. I have a lot of my advisor says I have a surgical hands. I still work with my hands. I'm not a sitting person, though I'm 70 years old. So hard work, I still work 70, 80 hours a week, and I run 10 miles every single day, and I eat once a day. So those are important. If you are not a good person, you can't accomplish anything. If you don't work hard, you have no right to complain. So disagreement is healthy. You can disagree and say what you think it is, but disrespect is not okay. You should not disrespect, even if you don't like it, even if you hate it. You should not disrespect. Then that tells you the character. Without a character, you can't be a good scientist. Take it from me. So I give you still, I give you a little bit introduction because it has some, I'm an official first batch when Zakir Hussain opened up Guntur PG Center, part of Andhra University, into Acharya Nagarjuna University. It's just a Nagarjuna University. Later on, it became Acharya Nagarjuna University in Guntur. And some of the plants we planted still there in 75. So I did a botany and medicinal plants and soil microbiology in 1977. I'm the worst student. Out of 30 students, my rank was 28. So, but I have problems. I have to blame P.V. Narasimha Rao, who is a relative to me, but he's the one who incorporated Telugu medium. So I'm a first batch of Telugu medium degree. And then when I came to master's, it's all English medium. I struggled so much. I couldn't write it. So that's part and part. And so when I came, so I failed in genetics. That was the part of the reason. And then when I came to US, my focus was two things. I need to take 4 or 5 genetic classes and get A, and speak English properly. Those were my first 4 or 5 years, of course, besides making money. I came to New York City my first year or 3/4 of a year. I worked in a drugstore in New York City in Manhattan, make $20,000, and then went to master's program in California. And that's where I took 4 straight genetics classes and got all straight A's. And that was an infectious disease. And when ciprofloxacin, which we abuse in India to prescribe, ciprofloxacin is a very quinolone compound. It's very, very important. We should not give as a first line because if you because ciprofloxacin has efficacy both on aerobic infections and anaerobic infections. So I'm the one. I have 6 or 7 publications before it is approved by FDA. And I went to Tucson, Arizona after I got married, did my PhD in tumor immunology because Arizona has a very high skin cancer because of the beta rays and desert nature in Arizona. And it has a chemo prevention program from NCI, a vitamin A chemo prevention program. And when I joined my PhD program, I went and talked to the director and said, I want to have a project. He said, get out of here. We are not interested looking at immunology. And nonetheless, after 2 years, I published 3 papers. Then I went and talked to him. I'm very sorry. I dismissing you 2 years ago. Okay, we will give you money. Show me. He gave me $50,000 and I showed. And then when they renewed the grant, I got $250,000 to finish my PhD. And what we showed is how vitamin A provides anti-cancer effects and through NK cells. That was my thesis and published a dozen papers. And went to Dartmouth and I did because continue to do, I have a reproductive immunology. Reproductive tract is a very fascinating mucosal immunology because it is physiologically flooded by the sex hormones, but yet protects from STD, sexually transmitted diseases. In a matter of it won't protect you just for 3 days, but rest of the month, it will protect females from STD. It was very fascinating work. I worked so hard, and I have again dozen papers. My first Journal of Immunology paper is from there. And then I went to, without an interview, I got an assistant professor job in a medical school in Chicago. And I taught for 2 years, both microbiology, medical microbiology, and immunology to medical students and pharmacy students full time. And then I went to Talvar Immunocontraceptive Company in Houston. Then went back to Dartmouth as a faculty. At that time, I have 3 children and I want them to show where I grew up in Guntur, and took them back to India and taught undergraduate students and master's students in Guntur, and then failed to convince a lot of doctors in Guntur, Vijayawada that we can do diagnostics, or didn't fail to convince, including Ratay Garu, that I could teach too well. And however, I was able to convince people at Harvard to get a faculty position at Harvard. Since then, since 2000, I have been at Harvard. I am an immuno-oncologist. I monitor last 25 years, couple of dozen clinical trials, how the immune system provides. We will talk about it as we go. You need to know a couple of things, CAR T, because CAR T is a EGNM. So you can't just sit through the EGNM and think about it. It's very easy. So you have to know a couple of things and see if I can give you 10 slides of my 40 years experience. A lot of people, I'm not a molecular biologist. I'm not a biochemist. I'm not an organic chemist. I am a basic cellular immunologist. I don't know certain aspects of chemical immunology or molecular immunology and stuff like that. But I know how the cells interact, how they talk to each other. I know as good as anybody in this country or anybody in the world. So immune system generates in flat bones, predominantly pelvic bones and the spinal cord, because those are the flat bones. Immune system consists of 4 different cell types: T cells, B cells, and NK cells, and monocytes. So B cells develop to certain nature, immature B cells in the bone marrow, and then they go to the either lymph node or spleen, and they will mature there. And the T cells, first 10 years of your life, will develop to some extent in bone marrow and go to the spleen, to the thymus, which sits right above your heart, and then it will shrink once you hit the puberty. It's no longer functional, but it does function 10%. NK cells also develop in thymus and in the bone marrow, and everything transfer after puberty to the bone marrow. And APC is also developed besides red blood cells in the bone marrow. There are 2 mechanisms in order to immune system to recognize. Those are the antigens called leukocyte antigens, what is called HLA, human leukocyte antigen. There are class 1, class 2. Class 1 is predominantly, there are 2 different T cells, what is called CD8, cytotoxic T cells, and there is another T cell called CD4 cell. It's like insurance company. It's a mediator. It doesn't do much. It will communicate with everybody. B cells, APC, and CD8 cells, which will kill the predominantly. So CD8 cells get presentation through class 1 restriction, and CD4 cells get presentation with class 2 restriction. And both class 1, class 2 predominantly express what we called antigen presenting cells, predominantly monocytes. Certain B cells and dendritic cells also express these molecules. In order to get a T cell, either CD8 T cell or a CD4 T cell, create antigen specific clone, they need to get 3 signals. First signal is this MHC restriction is with a peptide. So when a monocyte, either it's a viral antigen or a bacterial phagocytosis, and it will lysis and create a peptide, and that will bind to either class 2 or a class 1 and present it to a T cell via TCR, T cell receptor, which will consist of 5 chains of CD3 and then either CD8 or CD4 along with it. That is the first signal. And the second signal was to CD8 molecule binding to B7, B71 or B72, CD80 or CD86. And the third signal is either co-stimulatory molecules like CD40 ligand and OX40 ligand or 41BB is the third signal. Then only it will percolate, producing a lot of IL-2 and create 1 T cell to million T cells. They all can recognize one antigen and communicate what to do next. And you also have to know what is an antibody structure. Antibody consists of 2 heavy chains and then 2 light chains, and a form with a disulfide bonds. So they combine, and then there is one region is called a variable region, and the remaining 3 portions are called constant region, and the bottom portion is called FC portion. So antibody never go sit on FC portion. Antibody goes on antigen binding site. That was a big confusion when I gave a lecture in IIT Bombay. So in variable region, what we call, you heard the types epitope. Epitope is not part of antibody. Epitope is part of immunogen or an antigen. So it contains epitopes. Those epitopes bind inside this groove. What we call is a paratope or CDR3 regions. There are 3 of them. Complementary determining regions, 1, 2, 3. And the idiotypes are the outside of this region. So you have to know that when we're talking about, and also need to know what is infection versus disease. We will come to that. And there are 5 different antibodies. and IgA called alpha, and IgD, get delta, IgE, epsilon, and IgG, gamma, and IgM is mu. The difference is this is a pentamer. It can bind 10 different antigens. Like if it is a bacteria, it can bind to 10 different bacteria at one shot and neutralize them. And IgG is the largest antibody in the body, and it can also cross the placenta. So that way, during the pregnancy, baby is protected from the mother's IgG passing through the placenta, and baby is protected from the infection. IgE is important in allergy, particularly in India, against parasitic infections. IgD, we don't know really. It helps release or secretion of IgM. IgA is a mucosal antibody. It's predominantly present in all secretions, including saliva, in all mucosa, including tears, and all sorts of secretions. So each one has its own specific function. Within the IgG, there are 4 different subclasses. There are, because of the structural differences, you can see Ig3 has what we call the hinge region is much, much bigger. And the rest of them are similar to that nature. And I want to give you some sort of a definition. Infection means you are exposed. You didn't have any symptoms or clinical, what we call is prodrome, means you have a fever or a running nose or a red eyes or itching. Those are what we call prodrome or symptoms. So in, for example, CMV infection, you can detect. So if you do an antibody detection, that CMV is reactivated on a solid organ transplant, so we can detect that. Because once viral infections, you get it like CMV, herpes, they stay with you rest of your life. They won't go. They just go in dormancy. So they get reactivated. When a solid organ, because they give immunosuppressive drugs, CMV get reactivated. So we can measure. That means it is active. That doesn't mean that you have a disease. You won't have any symptoms. But if you have a symptoms, fever, malaise, or fatigue, lymphocytosis, 2 terms is cytosis means more lymphocytes. Tinea means reduction in the number. Those are the medical terminology you need to know. So when a disease means you have symptoms. So infection is different, disease is different. You have to understand that. The another concept in immunology you need to understand is primary response and a secondary response. This is where the vaccines work. When we give a vaccine, which is a primary response, the primary antibody, as I mentioned, is IgM. When you are exposed first time, either COVID or any bacterial infection, including sore throat, you get IgM production because, as I said, it has 10 arms to neutralize the infectious agent. That's why IgM goes up first. And there is in a B cell biology, when you exposed again, or you give a vaccine like a COVID. So when you actually exposed to the COVID, and then it will switch. In a B cell biology, when they mature in the lymph nodes, they class switch from IgM to IgG. And this is a log scale. We're talking about here 10 times, and we're talking about here 1,000 times. So it is 100-fold increase, and also it will switch from IgG to IgM. The major difference from all the antibodies and IgG survives in the blood more than a month before it degraded, whereas the rest of the antibodies is a week time. Less than a week, they will go off because again, they have to keep on producing. That's another advantage IgG have. You can ask, if we can produce antibodies, and antibodies can be used for the therapy. Yes, you can. So there are 3 different methods how antibodies work. There are ADCC, antigen and antibody-dependent cell cytotoxicity, antibody-dependent cell phagocytosis done by macrophages, and complement-dependent cytotoxicity. So ADCC, ADCP, CDC. Just show you quickly. So as I said, antibody molecule, the bottom portion is called FC. Antigen variable region is called antigen binding site in the funnel shape. So number of cells, including NK cells, antigen presenting cells, have a FC receptors. There are 10 of them. So FC receptors will bind, and then antigen goes and bind on a tumor cell. The antibody goes and bind, and antibody binds to NK cell, and this NK cell will release how they generally kill either cytotoxic cell or NK cells, will release what is called granzyme B or perforin, and they will make a holes on cell membrane of a tumor cell, and then everything leaked out, and that's how they die. And complement mediated, complement has a 9 different protein labeled as a C1 to C9, and they will make, once the complex is formed, they will make a MAC, which will make a pore on the cell membrane of a tumor once an antibody binds to the tumor cell antigen. And the last one is there are different FC receptors will bind to the monocyte, and they will phagocytize the tumor cell. So, and then once it is phagocytized, the lysosomal compartment will come and lyse the tumor cell. So 3 different ways, and we will tell you why that may not work in all the time. To just give you cancer has 2 important properties. One is cancer cell, because immune system, when it is developed, takes at least 4 years. In your first 4 years of life, immune system is not completely developed. That's why breastfeeding is important, because IgG pass through the milk, and baby gets lots of antibodies against infectious agent if a mother feeds baby with the breast milk. We'll have a protection, because immune system takes 4 years to develop in the baby, okay? And 40% of all of us are going to get in the lifetime cancer, because that is the risk of it. And the 2 features is cancer cells are nothing but proliferating cells. Like a skin, this is terminally differentiated cell, the top layer. All of a sudden, when you get a melanoma, this top cell will start dividing. You can give a number of reasons. We still 100% doesn't know what happens. We can give beta radiation, all sorts of things, lung cancer with smoking and all sorts of things. But why it is proliferating is still 100% we don't know. And the second part is, when immune system is developed, it is been shown that, hey, do not respond to the self-antigens. Your insulin or albumin or cellular protein do not respond. So autoantigens will not produce any immune response. Cancer cell is an autoantigen. So that is immune system caught in the middle. Should I respond? Should I don't respond? There is another disease called autoimmune disease, systemic SLE, systemic lupus erythematosus, or MS, myasthenia gravis, all sorts of autoimmune, rheumatoid arthritis in the joints. You can't move your knees. All those are autoimmune diseases. That means when what we call that showing the autoantigens don't respond is called tolerance. So you tolerize autoimmunity, so therefore you won't get autoimmune diseases. But when tolerance is broken, you will get an autoimmune disease. So cancer is a fine, idu antaru chusaru Telugulo, katti meeda samu. A fine katti meeda, if you are not careful, you will cut into 2 pieces, or if you fall this side, you fall that side. So that's where immune system caught into it, okay? So the treatment is predominantly surgery. Like, for example, colon polyps is intestinal lumen. You will have like a cauliflower type of growth in the intestine. So surgeon can go in and snip it off. So that way, that is easier. Chemotherapy, you can give high doses of chemotherapy, cisplatin, and all sorts of TNF alpha, but they all have side effects. They will also kill normal cells. In general, chemotherapy, most of the people die not because of the cancer, because of the treatment. Radiation therapy, which is you exposed to the radiation, which we worry about getting skin cancer, which is also important. But all of these treatments, even if you remove the colon polyps in the surgery, you don't know. When a surgeon goes to clip the colon polyp, he doesn't know where the colon polyp and where the normal, it's not visible to the naked eye. Hope that he removed all of the cancerous material. So you have to wait that polyp is not coming back at least 6 months, or a breast tumor. Remove somebody breast tumor. You don't know it won't come back. It will take 6 months to 1 year. It won't come back or not. So all these therapies currently used last 40 years will take time, anywhere 6 months to a year, to know that you have responded or did not respond. Though the disease that I work last 25 years is called multiple myeloma, which is a, in the B cells, the importance of the B cells is B cells produce antibodies. Before they produce antibodies, which I showed you, there are 5 different kinds of antibodies, IgG, IgM, and all. The B cell become a plasma cell. So let's say you have a sore throat, which is here, and your tonsils are right underneath in here, and that's where the antibodies are produced. As soon as they produce the antibodies, B cell become a plasma cell, and plasma cell start producing antibody. But that goes to a spinal cord, and that's where it will dump. All the plasma cells are made, they will go back to the bone marrow and produce the antibody. So most of those plasma cells producing antibody will not survive more than 6 months. Let's say you got a COVID shot and they produce antibodies. After 6 months, all the antibody plasma cells will die out. Who will survive is a memory compartment. A B cells and T cell memory compartment survive in the tissue, in the mucosal tissue. So whenever you re-expose, this memory cell become a plasma cell. That plasma cell go back to the bone marrow and produce antibodies again. So, however, in the multiple myeloma, that plasma cell become a tumor cell. It become malignant. It become, it never die. So, and then it will cause the bone damage. So this spinal cord become brittle. And then you bend, you have pain, all sorts of things. If it is pelvic, you will have a severe pain. If it is in knees, so that is the disease we work, and it is incurable. And with all the combinational chemotherapy, the therapies that I mentioned, we can only extend 5 or 10 years of life. And there are big people, and Dana-Farber Cancer Institute is #1 for the treatment. We have 30 physicians just to treat this disease in Dana-Farber. And so we are one of the best. So last 20 years, I showed in these patients, there is immune system suppressive dysfunctional nature of it. First one we showed is within the T cell compartment, there is a regulatory compartment, what is called regulatory T cells. Within the myeloma, there is a precursor disease like a colon polyps to colon adenocarcinoma, which is a malignant disease, whereas colon polyps is a benign. It doesn't transport it to lungs or liver or anywhere else. Same thing, myeloma is a bone disease. It will sit in the bone and destroy the bones eventually, and your bones become brittle and you break them very easily. So AMGAS is a monoclonal antibody, is high level, but it is not involved in the bone disease. So what we found is FOXP3 is a transcription factor expressed in regulatory T cells, and we measure that in normal, it is around 10%. within the CD4 compartment is 4% and other cells. Whereas in multiple myeloma patients, it is much, much lower. That means they are not present at high levels to control the immune responses. And the other one we showed is IL-17. IL-17 has a dual function. It is good against fungal infections, but it is detrimental, and also it's a pro-tumorous. IL-17 will help tumors to grow. So we showed that multiple myeloma, both in the blood and in the bone marrow, IL-17 producing cells are
Ragharu, your voice is not audible.
Yeah, just 1 second. The here internet get disconnected. Can you hear me now?
Yeah.
Can you hear
Yeah, we can hear you.
me now?
Voice is breaking. Voice is breaking.
Can you hear me?
Yeah, voice is
Okay.
breaking.
Let me see if I can log in back.
And
Is it okay now?
yeah.
Okay, let me see if I can share again. Is it okay now?
Yeah, it is being. Yes.
So we also did IL-17 antibody, which is a Novartis one, see if it works against myeloma. And this is blue line is without antibody treatment, and the red line is antibody treatment, prevent multiple myeloma. Novartis did not pursue to treat the multiple myeloma because FDA already approved to treat IL-17 antibody is called Cosentyx to treat rheumatoid arthritis. So that's a blockbuster for them. So they didn't pursue this aspect of it. And other cells, like there are 6 different dendritic cells. One is called plasmacytoid dendritic cells, also much higher in multiple myeloma, which will produce all sorts of interferons to screw up the immune system. And there is also suppressive compartment in myeloid cell compartment is called myeloid derived suppressive cells, which are also higher in multiple myeloma. So we showed that multiple myeloma already have immune suppression to begin with. So we have to take into consideration that aspect of it before we treat them. So going back to the why CARs are important, because immunotherapy and the CAR treatment is going to dominate. If you remember HIV or COVID last 3 years, how it dominated, CAR T cell therapy will dominate next 2 decades, because I will tell you reasons why. Okay, so immunotherapy consists of not only CAR T cells, but monoclonal antibodies that are being approved by FDA, bispecific antibodies, which can attack 2 different antigens at the same time. And the antibody conjugates, what they do is an antibody that can bind to an antigen on tumor cells, and they will put a conjugate, drug conjugate, which will, as soon as it binds to the antigen, it will be released and kill the tumor cell. And checkpoint inhibitors, there are inhibitors like PD-1, TIM3, TIGIT, and LAG3, which are the ones will tumor promotes, and so that way to suppress or evade the immune system, and we could produce the antibodies and also vaccinations. But except CAR T cells, all these cells will take time. Even an antibody, right? Antibody will go to the, let's say it's in the bone marrow tumor, antibody will go and bind. How does it kill? So T cell or other cells have to come there and bind their FC portion and then kill them. So that will take a time, and it will take, by the time antibody will disappear or it won't bind, all sorts of things. And vaccine will take a time, and checkpoint inhibitor, same thing. And we will talk about why CAR T cells are such a prominent. There are several types of CAR T cells, as somebody mentioned in the beginning. CAR T cells, father of CAR T cells is Carl June, who is educated at Harvard before he moved to Philadelphia. Carl June is a Harvard Medical School in his early part of his career, and I know him very well. He came to several times to give talks at Harvard. So there are primary reason is that, so now hopefully you will remember, so we use in the antibody, there are 2 heavy chains and 2 light chains. In the CAR construct, we only use single chain. So either single heavy chain, that's what those VLVH, variable region, that last portion where I showed the paratope or CDR 1, 2, 3 regions are, they take that portion and create a single chain and with a linker, and then there is a transmembrane. So this is served as an antibody. So you can't say CD19 CAR. That is not technically correct. It's anti-CD19 CAR. So it is an antibody. CAR is nothing but an antibody. So it is anti-CD19 CAR, anti-BCMA CAR. So anything is, they don't put anti, but technically it's anti whatever the antigen is, and then CAR T. Okay, so this is an antibody with a single chain. And this is different than the regular antibodies giving, is that they put it underneath signaling effects. So when it binds to the antigen on tumor cell, it will stimulate the T cell. I mentioned those 3 signals in the beginning of it. So one of them is CD3 zeta. So they put the CD3 and they also put the co-stimulatory molecules, including CD28 or 41BB. So this is the 2nd generation, and 3rd generation, they added multiple co-stimulatory molecules, and then they added cytokines and all sorts of it to improve CAR T generation. As I said in the beginning, CAR T production is an agony. It's not as simple as we talk about it. It takes 4 to 5 years to come up with that can work, and to do the phase 1, phase 2, phase 3 clinical trials to get FDA approval will take another 5 years. So this is a 10-year process in order to get a product out, to sell it and to treat the patients. The first 5 years is the critical part of it. There are a number of steps in the whole immunology involved, and also a little bit at the molecular biology in the last couple of steps. Identification of a tumor antigen. The identification of a tumor antigen is the most important part of it. It has to, tumor is expressing that antigen because it has a survival benefit. Otherwise, it won't affect. That is the only reason that will proliferate and become immortal. Then that antigen is important. If you look at it, why we selected BCMA to generate anti-BCMA CAR for myeloma is that there are 2 compartments. It does express very high level, both BCMA, and there is another CAR developed is called GPRC5D. They both are very high in myeloma cells. You can see these are primary cells. Double high is most of the very few patients express at low level, even if it is 20%. And it has a survival benefit. You can see if you take a 50% survival, that's a 2 years to 4 years survival, overall survival, or event-free survival. So it is associated with a survival. So it has to be an antigen in order to generate a CAR T, has to have some association in terms of the expression, in terms of survival benefit. And then you will determine, right? You take a sequence, like a BCMA is like, 400, 500 amino acid. That's a big, so you have to pick a portion that is important to bind to generate antibody. There are a number of software that takes almost 6 months to identify that, and it has to be extracellular portion of it. And then you generate that sequence of that antigen, hybridoma, like produce an antibody, and you will generate 10 to 20 clones, and you have to evaluate each and every clone against the antigen with the blocking assays, either ELISA or flow cytometry or Western blot. It takes another year to determine which clone you will go after. So to validate and sequence. So once you identify that out of those 20 clones of hybridoma clones, it will take sequencing that clone and then doing the DNA sequence. So that way you will know you need the nucleotide sequence, not the protein sequence. You need the nucleotide because you have to transcribe this antibody. So you get the nucleotide sequence, and then you get a vector and you clone this portion using the restriction enzymes and the ligases. So that way, this vector contains this particular single cell sequence. And then you propagate that vector. So that way you will have an amplifier. You could generate that. And then you transfer that into the mammalian system, because the cloning and amplification is done in the bacterial system. Usually use E. coli to amplify the vector. And then you bring it to 295 cells, a mammalian system, expression system, and then where you generate lentivirals containing this construct. That validation and determining the dose will take another year. And then you use those lentiviral transduce to T cells to generate anti-antigen CAR in a GMP facility. Building GMP facility, I've been struggling last 6 months in India. And so that will take, this whole process will take anywhere 3 years to 5 years. To do the clinical portion of phase 1, phase 2, phase 3 trials will take another 3 to 5 years. So following that is, there are 3 major steps. Once you generate, you validate the CAR and do the phase 1, phase 2, phase 3 trial, and FDA is approved, and then you give, how do you give? So first you need to draw 100 to 200 ml of blood from a cancer patient, what is called apheresis, and you separate the T cells and you freeze them and you transport them. And that's where you transduce in a GMP facility. First you activate those frozen T cells, thaw them and activate them, and then lentiviral transduction, and then you verify how much T cells got the CAR incorporation, and then you will determine the dose, and then you amplify, because you need to give anywhere to 100 million, because you can't separate the CARs. So you have to give the whole mixture, and you freeze it, and then you transport, and you have to check, make sure that, because it is developed even in GMP facility, make sure you are not incorporated any other viruses or bacteria. You have to do quality control. Anything that is going into the human body has to be very diligently have to evaluate, and then freeze them and transport them. And once you identify the patient who have given the blood, and he has to be prepared to infuse. Okay, so what they do is, this process here, even in the United States, will take anywhere from 1 to 3 months. And there is a 3-month waiting. This cancer patient is eligible to get a CAR T treatment, he has to wait 3 months. And the process takes 3 months. So 6 months, he's been waiting for the cancer. So you have to come up with the bridging therapy. Is that bridging therapy is going to affect CAR T therapy? You have to be mindful of that. That is where we are working right now. What bridging therapy would be useful for CAR T therapy to amplify or reducing the relapse rate. So that is very critical. On top of it, patient has to be treated with a lymphodepletion because patient has his own immune system. Even though these are autologous cells, but there won't be much room for CAR T cells to expand once you are given to the patient. So you have to remove all patient immune cells. So that way, when you give CAR T cells, it has ample room to expand. But it will also cause, there are so much T cells dividing so fast, it will cause complications and toxicities. So bridging therapy and lymphodepletion is the two things you have to prepare the patient before you infuse the CAR T. Okay? And CAR T cells, all this manufacturing, lots of people do it manually, but there are 3 companies make automated. So from step 1 to the infusion part and characterizing up to here, completely automated. So those are the 3 companies. First one is Miltenyi Clinimax Prodigy, which cost in India somewhere around 2.5 crores, just this machine. And what it does is you bring the leukopak here, the apheresis, and then it will generate CAR T cells by step. You just have to change media and all the supplements. You change the bags, you wash them, you put new bags, and it will make. You are not manipulating manually. There is another machine by Lanzo. Cytiva is the another one called Sophia, which is also cost. I don't know whether it is available in India. It costs same thing here, $200,000, $300,000. And there's the last one is Lanzo, which is called Cocoon, which is also same $200,000, $300,000 here in, these are all automated systems to produce CAR T. So there is no manual, you don't produce manually incorporating what I showed you, which will take 3 years to manufacture and certain parts of it. So far, even in this country, there are 7 CAR T therapies are approved. And 2 of them are for multiple myeloma, BCMA based, BMS, Bristol Myers Squibb has Abecma, and which we worked on it when it's inception, and Carvykti, which is the difference between both is, as I said, these are single chain, single heavy chain, single light chain is in Abecma, whereas in Carvykti, instead of putting heavy chain, light chain, they put 2 heavy chains. So that is the difference between that. And Carvykti has a slightly better response in terms of both of these approved by FDA. There are CD19 for leukemia and lymphoma. There are 4 of them approved from different companies, Kite, Novartis, and BMS. There is a recently approved couple of months ago, there is a solid tumor. This is where this is a restricted, class 1 restricted, a maize A4 antigen, TCR based. It is a UK company called Adaptive Immune. This is for synovial sarcoma. And there are also 2 bispecific antibodies approved last month here by FDA for solid tumors. And so it's coming. Predominantly, it has been shown only liquid tumors, CAR T cells, but solid tumors are coming. So why CAR T is going to dominate next 2 weeks compared to traditional surgery or combinational chemotherapy or radiation therapy for cancer has been given last 40, 50 years, because none of those therapies will give you a response less than 2 weeks. They will take a year. Whereas CAR T, you will see within 10 days, complete response, or you will see a response, and within a month, you will see a complete response. That is the magic bullet. So that is what the patients want. In some cases, it will sustain for 3 to 5 years without a relapse. And we have seen that. So just for that response, quick, robust response, though it involves in time consuming and all sorts of other things, this is going to dominate. So that is number one reason. Does it have drawbacks? Yes, it does. One is the time. It takes 6 months to get a CAR T treatment into the patient. It may take longer in India. And it is an expensive proposition. Even in this country, it is $500,000 plus hospital costs. India, there is, I will show you, and it is 3 months waiting, even in this country. And another drawback is eventually, not everybody will get 5-6 years without relapse. At least 50 or more will relapse eventually. And there are cytotoxicity, CRS, which you all familiar with COVID, cytokine release syndrome. So number of cytokines, because CAR T cells, as soon as they are infused into the human body, and they will go directly into the circulation and go into where the tumor is, and it binds to the tumor cell and kill them so rapid. They release so much content of cytokines, it will affect all the other aspects of normal tissue. So that's why you will have fever and cytokine-induced or release syndrome, and also neurotoxicity. So you will have a less sensation or tingling and bothering neuropathy. So those are very common. And we have to report FDA if they are beyond stage 3, both CRS and neuropathy. And if it is beyond stage 3, you have to stop the therapy. And they are given for cytokine release syndrome, they give anti-IL-6 immediately when fever spikes. So that is additional cost. So there are drawbacks, but there is a benefit. And if a patient, especially in myeloma, you are destined to die because there is no cure. We can only extend 5 years. But if a CAR T will eliminate the disease and I have a chance, maybe cure, they will take that. In India, there are 4 companies or 3 companies and 1 academic institution. Immuno-Act is the only one with the Tata, has a CD19, anti-CD19 CAR to treat the patient. I don't know, I can't get exactly, they said they want to treat 1,000 to 1,200 patients. So that's not possible, at least it's not in a realistic fashion, because there is not an automated CAR production.
So they have so far treated 100 patients. That was the recent announcement.
Right, that's 100 months since its approval in late October of last year. That's almost more than 8 months. The projection was 1,000 to 1,200 patients. So that tells you how difficult it is, not only getting the hospitals involved and recognizing the patient, but also even if you have patients, to generate the CAR, even 24/7, if it is not an automated system, it is a difficult task. With automated system is like that Milteni, you can only treat 3 patients per month with 1 machine. So because it takes a week to generate within the automated machine. So For transportation to get the blood sample, transportation to the car will take at least another week if it is not in the same place or in the same nearby. So it is not, but that's company is good. It's a good because of Tata. They were able to do it.
Is
And
is it Laurus or Tata?
it is Tata. I think
Yeah,
Chandrasekhar.
I remember that there was a big announcement by Laurus and Immuno-Act.
Right, but they may have because they are trying to raise
maybe more than one source.
right. Because Immuneal raised 100 crores just the last couple of months from Arabian countries because she is known. Dr. Shah is a very established pharma biocon company, and she built an excellent facility in Bangalore, and they are association with Spanish, um, and they are still doing phase 2, phase 3 trial and hasn't gotten approval yet. Again, that is also anti-CD19 CAR for leukemia and lymphoma. And Vikram Matthews in CMC Vellore is the Miltenyi with using Miltenyi CAR and treated and got I think very close to getting CEDESCO approval pretty soon. And so that way he can treat in Vellore. And Origin in Bangalore is also the only one. All three of them are anti-CD19 CAR, and Origin is developing a BCMA CAR.
So Origin has established a facility in Hyderabad.
Right.
And probably they will take up here in Hyderabad.
Right. That's a part of Reddy Labs.
Yeah.
But Reddy Labs has extremely well known for the therapeutic drugs, but not cellular therapies. So we have to wait and see. But so the Immuno-Act is selling somewhere around $45,000 to $50,000 or lakhs, however you say that, and plus hospital costs, like I told you, to do the apheresis and to do the bridging therapy, to do the lymphodepletion, to do the CRS controlling antibody treatment. And you have to stay minimum a week for infusion of the CAR T, hospital stay. And then also all those you combine easily comes another $50,000 or 50 lakhs. So we're talking about even in India, low cost manufacturing still costs you 1 crore. Whereas if it is $1 million here, that's 8 to 10 crores in India. So cost is another aspect of a drawback. So what I focus is, like I said, is one aspect of it. Not only make as cheap as possible compared to $1 million, but my primary thirst is, could we identify these guys as rapidly as possible? Who is going to relapse? How do we treat them? How do we make them sustain CAR T response? So this is what been last 5-6 years. We have been very vigorously, robustly working on how do we complement CAR T therapy to make it sustain much longer. So all the B cell lymphomas coming from B cell development in the bone marrow. There are a number of pre-B cells, immature B cells, Burkitt's lymphoma, follicular diffusing lymphoma, mantle lymphoma, multiple myeloma, Waldenstrom, all these diseases in the development of B cell compartment. That is where they generate. So you have to know that. And this is where we're focusing. We know CAR T will give you excellent response, but there are also relapses, less than a year. How do we improve that? Is there a particular bridging therapy will help sustain the CAR T therapy? is a particular lymphodepletion will help? And is there any particular therapy we can supplement or complement type of a thing? And is there any other faster, cheaper CAR-T manufacturing? Can we do it fresh compared to frozen? Or can we do it within a week? Once a patient gives the blood, could we inject him back within a week? So those are the ones my focus is last 6-7 years. I will show you just brief data in those type of things. So these are, we do a single cell sequencing in the lab, what is called site-seq, which is a very important. And the left-hand side panel is showing a responded patient for CAR-T therapy, and the right-hand side is a non-responder. When we look at it, and then you can cluster them using all sorts of computer programs, and we can tell them what cells they are. And these are monocytes, these are lymphocytes. And you could see unresponded patient, just a visual. There are a lot more monocytes here compared to responded patients. That is right there is telling you there's something wrong in the cellular composition. And when you look at it, I told you the CAR-T killing tumor cells, there are two cytokines involved. One is called granzyme B and a perforin, which once poke the holes into the cell membrane of a tumor cell. They are much higher level. This is at 1 month of the infusion of CAR-T, and then this is 3 months after the infusion of CAR-T. And these cells still capable of producing these cytokines and kill the tumor cells if they come up. Whereas non-responded patient responded very slow level and disappear. So if we can identify this as quickly as possible, and then we can think about how do we treat these non-responded CAR-T therapy patients with what? So another example, we look at it, their B cell receptor. There are VDJ region, variable region, diversity region, and J-chain regions are important. How diversify. This is where one of the guy got a Nobel Prize, telling we have more than 1 crore trillion combinations we can generate in the human body. So you can see this is a healthy, how robust the variability or diversity of that receptor composition is, and compared to myeloma patient, and how sparse that is. So we're trying to identify what receptors that are not coming and how do we amplify that. We do a site-off. Generally, there are 3 methods: flow cytometry, which is lasers using and conjugated antibodies with various colors, FITC, phycoerythrin, tandem dyes. Hello? Tandem dyes, so that they will have a compensation issues, laser going onto those dyes, whereas site-off is based on mass spectrum, because it's a mass molecular nature of the metal. We conjugate to the antibodies. It's a sort of a black and white, very easy, and doesn't have a compensation issues. And also you can do 40 markers in a single cell. So when you do it, healthy bone marrow cells, you can easily tell monocytes, NK cells, CD8 cells, CD4 cells, B cells, whereas in CAR-T treated patient, they're all over the place. As I shown you, there are high monocyte compartment in one patient, and there are disturbed and very few NK cells and very few B cells. So even in a visual fashion, you can see it very quickly. And that is what we are trying to identify, use these technologies to recognize how do we do it. So I'll show you another example. The top panel patient relapsed within 3 months of the CAR-T therapy. This patient sustained 15 months, 1.5 years. And we try to look at it. CAR-T, they both have a very high CAR-T. The blue ones are CAR T within the T cell compartment. And the green ones are CD4 and the red ones are CD8 here. And what are they expressing? In here, they're expressing inhibitory markers like a TIM3, whereas responded patient does not express them. So can we treat this patient with anti-TIM3 along with it? How soon we can start? So stuff like that will give. So technologies like CyTOF, flow cytometry, you can only do maximum 7 or 8 colors. 8 markers you can determine on a given cell, whereas CyTOF, you can do 40 markers, 40 cell surface markers. That is what we do. In CytSeq, you can do 200 markers. So that is what I showed you in the beginning. So CyTOF will also give you functional nature of it, not just one cytokine. We can measure 20 cytokines. So that way, that will tell you the functional ability of those cells. And are they combinational? This is total T cell compartment, and these blue ones are CAR T cells within the T cell compartment, and these red ones are those CAR T cells producing TNF alpha, and those CAR T cells producing this orange one is interferon gamma, in addition to TNF alpha, and GM-CSF in green, only a portion of it, producing these triple cytokine, and they're also producing granzyme B, these dark green, and they're all CD8 cells. So we can identify, is this population diminishing or how do we improve those? So another example was, you can see it very easily. These are CAR positive and interferon positive, IL-2 positive, and these are CD8. So it's very easily, we can tell a combinational immunotherapy. And we can also do a various subsets, memory compartments within the CD4, effector memory, naive cells, central memory, exhausted cells within the CD8 very easily, and by flow cytometry or from CytSeq. And various other subsets of B cell compartment, B1 cells, B2 cells, regulatory B cells, and cells that are gone and mature in germinal centers, and all sorts of other things. And NK cells, various inhibitory markers that they are expressing on NK cells, and are they associating with survival? Higher expression of those will have a better survival. And so these are the ones who will give us complementary, come up with the new regimens to complement CAR T therapies, which we have, and we are in the process of utilizing those. Okay, so thank you very much. If you have any questions, I'm more than happy to answer.
Thank you, sir. That was a very interesting and comprehensive talk. We appreciate you took the time to come out here, and most of the people on this call, they are students and will have many questions for you. You guys can raise your hands and start asking, or type in the chat box and I'll read them out.
So this is Srinivas. Very good, interesting, engaging talk, sir. So I have one question.
Yeah.
The IL-17, that is like Janus-based cytokine,
Right.
especially it has a positive and negative effects,
Correct.
right? So
Correct.
people having psoriasis, right? IL-17 is expressed predominantly.
Right.
But the same one that can lead to some sort of tumor or cancer in the psoriasis patients?
I don't know. I'm not fully aware of that literature, whether psoriasis has a cancer complications or not. But it's only been what L17 that Cosentyx is only approved last 3 or 4 years. Anti-L17 antibody. There are 3 companies got approved to treat rheumatoid arthritis and psoriasis. It will take a little longer to show whether those anti-IL-17 antibody treated patients have a cancer incidence or not. It will take a little longer But it has been shown in animal models, IL-17 is a pro-tumor. It will help tumors to grow. So that is a little caveat.
Thank you, sir.
Chirag, you want to go next? Unmute yourself, please.
Hello.
Yes.
Sir, Chirag Gala here. Sir, I want to ask, sir, if this CAR T cell will be more effective or that bispecific T cell engager, because both are dealing with the T cell for providing the effect to cancer. Like,
Right. So in terms of the cost-wise, they both are more or less same because antibodies and bispecific, it's not a one-shot deal, right? In a CAR T, it's a one-shot deal. You just get one infusion and that's it. Whereas bispecific antibodies, you have to continuously get. That's one, even if they have similar efficacy in terms of the response and all, expense-wise, it is same. And the toxicities are same, CRS, neurotoxicity, neuropathy, all those are same. But as an immunology perspective, I told you, antibodies take little longer. They're not equipped everything in them. They need a help. T cell has to or NK cell has to bind their FC portion and then kill. Whereas CAR T is like a one boatload. It has everything. It has antibody that binds to the antigen, and it has signaling molecule and activate the T cell right there and then. And so it is done. If it is relapse occur, it will occur in both cases. Price-wise, it's same. Antibody may be little faster to produce because it's off the shelf. So that's only the advantage with the bispecific because it's ready to go. Maybe you don't have to wait. But expense-wise and long shots, because you have to take continuously every 3 months number of cycles. So antibody can be used, bispecifics can be used. So that's why some of those cases right now, last month, for some solid tumor, there is a bispecific antibody is approved for EGFR mutations and MET mutations. Bispecific antibody is approved by FDA to treat some of the lung cancer patients.
We can go with Anagha. Anagha, unmute yourself.
Sir, do this theory is only useful for myeloma cancer or it can be useful for another purpose also?
No, each myeloma, we're only using anti-BCMA CAR, and anti-CD19 is for leukemia and lymphoma. So solid tumors are coming up. So you have to make individual and also individual patient. So that's another cumbersome part of it. So you have to, CAR T, you have to make for every patient, you have to make for them. Whereas bispecific, it's off the shelf.
We can go with Madhavi next. Please unmute yourself.
Yeah, sure. Yeah. So thank you very much, sir. It's very refreshing immunology background and nice discussion. So this is a basic question regarding what is the average life expectancy of a patient after this therapy?
Generally, what we have seen, I can only talk about multiple myeloma, anywhere from 3 to 5 years without a remission, and beyond that is difficult to predict. We need some more time to have. What I meant is that quality of life is much superior with the CAR T therapy compared to other therapies.
Okay. Thank you, sir.
Sir, I'm Professor Krupanidhi. What about the half-life of CAR T cells, sir? Do they stay forever or 4 to 5 years like this?
The persistency hasn't been associated with the response. So whether they stay 4 months, 6 months, generally they won't stick around more than a month or two because, right, there are two processes involved. One is when any cell that divides continuously, particularly in the immune compartment, what is called activation-induced apoptosis. So upon certain cycles of activation, they will kill themselves. That's a built-in process. So T cells, they will die off because after 5 rounds, 10 rounds of proliferation. The second aspect of it, there is like I showed you in the memory compartment. The effector memory cells are the ones very effective killing tumor cells. And if they do 2 or 3 rounds of killing without killing themselves, they will become exhausted. They're no longer able to kill and they will die off easily. And also they have a half-life, right? 2 months. Generally, T cells only survive 60 to 70 days. So we're talking about 2 months. But there are memory cells can survive longer than 2 months. If a CAR T cell become a central memory cell, it can survive longer and protect. That's what we saw in some of these patients who have not relapsed for 2 years, 3 years, 4 years, is their memory cells are still there. Even though CAR T, we can't measure that many CAR T cells, but if we take them and activate them in vitro, they respond.
With regard to the
another
plasma,
side.
with regard to the plasma cells are regularly renewing, no?
Plasma cells are programmed to die within 6 months.
But
Once they
new plasma cells are again renewing, sir.
right from the memory compartment.
Yeah. So they also will be having the cancerous antigens, no? Upcoming cells.
Myeloma is an immortal cancer plasma cell, right? It's already malignant.
Oh.
Are we killing all of them? Or there is a little bit that, there is never say never 100% guarantee, right? There may be 1 or 2 cells alive and they are coming up. Usually that is what the case is so far.
So that means they restore the humoral immunity, sir, once the treatment is done.
Correct.
Yeah. Thank you, sir.
Sir, another side, while we are constructing the CAR T cells, we have discussed that we are taking one heavy chain. So my question was, as there are two sections, constant and variable, if we would be taking the variable region, it would be more specific. And if we can't do this thing, what is the role of constant region in the construction of CAR T cells?
No, we don't take a constant region. In the CAR T I mentioned, it's only variable region. Even within the variable region, I showed you where the CDR complementary determining regions where they bind to the antigen. There's only so much portion bind, and we sequence that part of it and only take 20 nucleotide, a peptide. We don't take any constant portions of it. The usefulness of the constant region is, like I mentioned, there is antibody-dependent killing mechanisms. So how do they work is through the FC portion, which has monocytes, macrophages, dendritic cells, B cells, NK cells have FC receptors, and CD8 cells have FC receptors. So they can bind to antibody through the FC receptor and kill tumor cells or infectious agents. So FC portion is only, constant region is only useful the stability of the antibody and also other uses. But we don't take constant region at all, even complete variable region. We only take 20 nucleotide worth in the middle of that groove where CDR3 regions are in the variable region only to generate only single chain CAR-T.
Minurani?
Okay, thank you so much.
Yeah, sir, good evening, sir. Sir, this is Meenu Rani here. And
Yes.
sir, I wanted to ask, actually, my question is two-part. First of all, I would like to say thank you. It was an excellent talk and it was very informative. Sir, my first part of my question is, sir, what are the, after the dosing, after the CAR-T infusion, so what are the precautionary things, precautionary medications or precautionary diet or something that the patient has to be in? And the second part is, have you seen with if there is something like that, have you seen that does it contribute to the expansion of the that life expectancy of the person more than that of the 5 years with addition of those things?
Right. The two side effects which I mentioned, CRS and neuropathy or neurotoxicity, are expected within a week, anywhere from 3 days to 6 or 7 days. So cytokine storm is predominantly symptomatic with the fever raising. That's why they have to be hospitalized for a week or 10 days, starting from the infusion day for a week or 10 days. So that way they can monitor. So if they see fever and CRS, which can be measured in the plasma within a day, what cytokines are elevated, how high they are. So immediately they give anti-IL-6 antibody therapy, which will also cost 20,000. So there are treatments ready to go, and neuropathy also have treatments in general. So those treatments are given. Those have a less bearing on the proliferation because it's a sort of because it's in a particular niche in the tumor, because the CAR T cells accumulate where the tumor is. So to all these therapies, usually, so right, we take an antibiotic to reach the tissue. Antibiotic serum levels and tissue levels are absolutely different, and their half-lives are different. So usually won't affect, and we can't say high CRS have a better survival, low CRS has a low survival, something like that. We can't say it at this moment.
Thank you, sir.
Okay, we have a question from Sandeep in the chat box. Can we engineer human T cells in such a way that it secretes short chain variable fragments or antagonist proteins against pro-inflammatory cytokines such as IL-1, IL-6 receptor alpha, TNF alpha, or interferon gamma, so that infused CAR T cells autonomously regulate inflammatory cytokine production to mitigate cytokine release syndrome?
Right. Theoretically, yes, but like right now we only give IL-6 because those are very inflammatory. IL-1 and IL-6 are very inflammatory if they cross the threshold level. So that's why we give anti-IL-6 antibody, which calms down the CRS. But it is not just IL-6 or IL-1 or TNF alpha. There is a battery or a spectrum of half a dozen to dozen cytokines. Which cytokine you will put it in? Cytokine is not the theory, or at least people have tried, but there is a receptors. So there is a CAR getting approved IL-6 receptor antibody. So it will bind. So what happens is, right, you produce the IL-6, it has to go and bind in order to make the receptor expressing cell get excited. And IL-6 go and bind to the receptor, and the cell will jump up and down. But if you add the anti-receptor antibody, so it will bind to the receptor, and IL-6 will not have a chance to. So they are trying receptor antibodies for TGF-beta and IL-6 and other cytokines. But it will also make you susceptible for infections. So that is another danger is involved, because we remove T cells, lymphodepletion, and CAR T cells are proliferating, means threshold is very high against only one tumor antigen. In case if you get an infection during that time, you will be very susceptible. So if a inflammation is completely stopped, that will increase your risk to get hospital-based infections. So people worry what prophylaxis, and those are monumental. If you get sepsis, you will die within 24 hours. So there are theoretically possible, practically they haven't been worked out yet.
We have a question from Mahinder, Dr. Mahinder.
Yes, yes. Thank you so much for the nice presentation. I have two questions. First question is the CAR T cell therapy, the cells need to be extracted from the patient or any donor can donate cells for the engineering? And the second question is, what is the production cost, or can it be massively produced, or like, how long it takes to produce these cells?
Yeah, it's a individual patient based. Each patient has to donate the blood in order to generate, otherwise it is allogenic, so they will reject. So it has to be autologous. So that's the drawback. Every patient has to give a blood and then it is generated. It will take anywhere from 3 months to 6 months to generate. And the cost is to manufacturing itself, even in India, is 40 to 50 lakhs, not counting hospital costs. That will be another reason, 40, 50 lakhs.
Thank you.
We have a question from Dr. Narsaya. Could you comment on secondary malignancies? Is there a relationship between secondary malignancies and relapsed patients?
Yes. So B cell CD19 CAR is been longer than BCMA CAR, and they are slowly. There are some reports that these patients may be susceptible a little bit to the secondary tumors, but it hasn't been conclusive. Okay.
And then one question from Dr. Mujib. There are many companies with ongoing CAR-T programs from existing landscape. What is the most promising CAR-T technology and why?
Because of individual patient involvement, automation is the critical part of it. If you don't automate the CAR manufacturing, it will cause tremendous problems and delays. It takes only small step go wrong, and you have to abort the whole process and start all over again. So automated CAR manufacturing is the way to go, but still it's expensive.
We have last two questions. One from Mohammad Jamil. Are there any success stories or clinical trial results that highlight the efficacy of CAR T cell therapy? And the last question is, would designing CD19, CD20 tandem bispecific CAR T cells offer persistent advantage over regular CD19 CAR T cells? Will it address antigenic escape issue in tumor and make bispecific CAR T cells more durable?
Right. So the first question is it is expensive. There is no question about it. And there is a boatload of evidence. That is why the momentum and CAR T popularity is that the robust response within a week. Cancer patient feels so happy. Yes, after 2 weeks, he feels like he is a new person. So that is the fascination or jubilation, whatever you call it. That's why immunotherapy, CAR T therapy is here. Besides all the toxicities and expensive and manufacturing time, it's going to be dominating next 2 decades, not just a day or two. The second part is dual CARs are right. The transduction, higher the length, higher the load is less the efficiency, right? That's a simple math. More length you add, less efficient, less, CAR T production, manufacturing failures, all these kinds. And the initial trials with both CD19 and CD20 in the same CAR did not show any improvement against single CAR of CD20 or CD19. And now we find out there are some issues involved because CD19 CAR is much more robust than CD20 CAR. CD20 CAR is slow, so it dominates, and CD20 doesn't play a role when it is a dual CAR. So we are figuring that out, and we just found out a couple of things which I can't mention. So there will be a, but it is a consensus not to do dual, triple CARs. You do multiple tandem CARs. So you generate CD19 CAR, CD20 CAR, and inject them at the same time. So that is much more preferable than putting CD19 and 20 on the same CAR construct and making express on the same T cell is not any beneficiary. As a matter of fact, it may be detrimental. So those CARs are coming, and we just have to figure out their structural role. CD20 is not well studied. The CD19 is extremely well studied. We know everything about CD19. We only know 10% of it. Why CD20 is there? So now we know there are 2 or 3 groups have some ideas. How do we overcome? How do we make CD19, CD20 dual tandem CAR rather than bispecific CAR? Maybe more useful. That may be coming in a year or 2.
Thank you very much, Dr. Rao. Can we squeeze in one more last question?
Oh, hello,
Sure,
ma'am.
sure.
I have one question. Can I ask you?
Yes, please.
Hello, sir. Very nice presentation. So, sir, I have a question. I have 2 questions. So, first question is that, what is the right time to start the CAR therapy for the patient? Because the cancer already detected in the very late stage. So, first stage itself is good for to start the CAR therapy. And the second question is that, does this therapy is equally good for the drug resistant patient, and so what's the efficacy for the drug resistant patients?
Yeah. So initially, I only speak on myeloma. So myeloma, when it is approved, those 2 CARs that are approved initially in 2021, they were approved only after 3 or 4 treatment failures on myeloma patient, then they are eligible to treat with a CAR T. And so it works. That is what the wonder is. It works excellent on drug resistant, which is what it is. Now, just beginning of this year, both same drugs are approved to give as a first line without a drug treatment because of its robust response. And on top of it, solid tumors, I think it's going to revolutionize because it doesn't matter whether you have stage 1 breast cancer, stage 2 lung cancer, stage 3 liver cancer, stage 4 lung cancer. CAR T will kill them. No matter what stage you are, they will be dead. Will they relapse? That's the question. So it will kill them compared to any other chemotherapy. So that's once that come into the play into the solid tumors, CAR T therapy will take linear leap into the cancer treatment. That is coming. There will be at least half a dozen solid tumor therapies will be approved by FDA in next year or 2. So stage malignancy won't matter for CAR T. CAR T will go to the tumor wherever it is, it will kill them immediately. So that is the beauty of it.
And sir, if patient like, because there is a time lag between when we collect the patient blood and we then make the CARs from their blood, and so there is a time lag between the treatment. So if so, the patient is, I think, already in the some kind of medication. So does medication affect the efficacy of this CAR treatment?
Right. So that is where we are working. So what we call is called bridging therapy. So once you collect the blood, what bridging therapy you will incorporate on that patient, which will benefit the CAR T therapy. So we are doing, lots of people are doing trials, and we have data because we analyzed if you give X1 bridging therapy, what happens to the CAR T therapy? If you go X2 therapy, what happens to the CAR X3, X4, X5? We have data to suggest which therapy, bridging therapy is better for CAR T. So I can't render those results, but they are coming. So yes, you have to be cognizant or aware of what bridging therapy you give. So that way that won't interfere with the CAR T or diminish it, CAR T therapy efficacy.
Okay, thank you, sir.
Thank you.
I cannot hear you, Deepika. You're on mute.
Good to see Dr.
Yeah.
Suresh in colors, in all colors.
Yeah.
Sir, I think you can give a closing remarks.
Yes, I think we invite Dr. Suresh Pusala, a science strategist, thought leader, and investor, founder of Acastra Health and Oncoseek Bio Private Limited. He himself is a very big researcher in the cancer field. Sir, please give the closing remarks and close the session, sir. And thank you very much, Dr. Rao, for spending this time with us and extensively discussing the research work that you did and also addressing all the questions. Thank you very much.
So it is the most comprehensive and authoritative presentation. Dr. Rao, congratulations, and really thank you for sparing your time.
Yes. Should I say a few words? Deepika?
Yeah. Please, please.
Okay. Thank you, Professor Redana and Srinivas Reddy Garu from Baltimore, US FABA chapter. I first of all thank FABA for doing this wonderful session on CAR T, which they have done many sessions. And I think this one, as Professor Redana pointed out, it's very authoritative, almost to the speed of what's happening in CAR T today, not what happened last month or 2 months back, because the field is moving fast. And Dr. Prabhala Rao, who is at a very prestigious institute, Dana-Farber, I'm familiar with the cancer research and everything that comes out from Harvard, Dana-Farber. So myself being a mucosal immunologist and having worked in St. George Children's Hospital for childhood cancer in Memphis, Dr. Rao, I can tell that it's a very challenging field. And for so long, we did not have any therapeutics for cancer. But my last 10 years in BMS, Bristol Myers in Redwood City, and also in India, where we built the Syngene facility, I can tell you that this field has grown in leaps and bounds. First of all, the last 20 years was dominated by monoclonal antibody therapies, which we continue to use, especially for PD-L1, PD-1, CTLA-4, all these checkpoint inhibitors that I was a part of the team. I can tell that when CAR T comes and adapts itself to treating solid cancers, which is the biggest challenge right now, because right now we are able to treat myelomas and blood cancers, but not solid cancers. But Dr. Rao, your talk actually gave me a lot of hope, and your vision and confidence in saying that in the next 1 year, FDA is going to approve 6 or 7 solid cancers. That makes me feel really, really stunningly surprised and also excited. So I'm looking forward to those things, and I only want to know that you are in the midst of all these things. And as Professor Redana said, that there is a lot of work being done in India as well, especially around Immuno-Act and Laurus collaboration and Immunil, also the Origin that is happening to working, and Dr. Matthews from Vellore. I think all these are good, but I think the microenvironment, the tumor microenvironment in the solid cancers is going to be a big challenge, especially the solid tumors with the necrotic centers and the surrounding capsule, if it is solid, malignant, but encapsulated, but spreading fast in multiple sites like papillomas in the colon. I think these are going to be very challenging compared to other liquid tissue tumors that could be a little bit more diffusely present. For instance, pancreatic could be much more easy to treat than a colon, or lung could be easier to treat than liver. So I think there will be a lot of organ differences between solid tumors. And I'm looking forward to your statement coming true. And your talk today gave us a lot of deep insights into the science, especially CD19, which is the dominant one right now, and CD20, which camouflages that. But then there were excellent questions that Deepika moderated. Excellent questions. Actually, I'm impressed with so many good questions, especially around the field of why can't antibodies against IL-6 be co-mixed along with the tumor cell, along with the CAR T cell and so on. I would like to hear one comment from you. This was my question. I should have asked it you in the question session, Dr. Rao, but I would like to ask it now. There are quite a few companies working on NK cell mediated CAR T therapy and other B cell and T cell and every other cell type. Of all these immune cells, and I do want to let you know that I have a PhD in immunology, but I never understood the thymus shrinking at that age when all the selection of immunocytes, lymphocytes, everything has happened, and letting the body take it over for the next 60 years or 100 years. And then comes the memory. Then you hit on all these topics. In your view, what would be the, especially just please focus on NK cells and other cells, and what kind of challenges it would have in treating solid cancers where the microenvironment and the tumor macroenvironment, which is capsule, fibrosis, collective tissue, blood vessels, and so on. What kind of challenges do you expect to see? Because you also said that there's going to be drugs approved in the next 1 or 2 years. And what kind of challenges have been addressed already that makes you come out with such confidence? And I really appreciate your answer for this. And you don't have to reveal all the secrets, but a little bit of insight would excite my scientific brain. Thank you, Dr. Prabhala, but excellent presentation. I rest my case, let you answer the question. Have a wonderful day. And all of you, happy Vinayak Chaturthi. And Professor Edena, you started a beautiful education session. And Ganesh Chaturthi, which is known for knowledge, right? So thanks for that, Dr. Rao, and you're an excellent scientist and very proud of what you have achieved. Please answer that question, and please convey the bye on our behalf as well to the team. I remain. Thank you very much.
Yes, Suresh.
I don't see Dr. Rao online. I think he logged off.
Dr. Suresh, so we need to maybe join hands in bringing him to India and establish the facility. And he is adamant on setting up the facility in Vijayawada. Let us focus. We will be very happy to help him. And I request you also to join hands and other people also to bring in and see that it is established at the earliest in India.
Sure, sir. So Deepika, can you please jot down my comments that I made and appreciate Dr. Rao
Sure,
on the
sure,
email?
definitely. The session is also recorded.
It's being recorded, so we can capture it.
Thanks,
so thank you very much.
Do you guys think we can take a snapshot of all of us if you turn on the webcams, please?
Yeah, everyone is.
I'm the one looking more colorful.
All of them have already left, most of them.
Only
but
the people
that's
are there.
good.
But still,
but I think most important is the speaker.
Exactly.
Yeah, I think that is the first thing that we should do next time.
I have taken a couple of shots in the beginning.
Sure, yeah, that should be good. That should be good.
I think before he starts, we should do that.
Yeah.
Or somewhere in the middle.
So now you're missing my colorful shirt in the first shot.
Yeah, you're still in US?
Yeah, I'm still in Austin, Texas, in my daughter's house.
Okay. When are you coming back?
October, middle of October.
Okay.
I'm
we
doing
look forward to it.
Sure,
Yeah.
Professor Redden. Yeah.
Yeah,
I
I took
suggest
the
that you speak to him.
Okay. I will.
Yeah, and then let us discuss on that.
Sure.
Whatever it
sure,
is.
sure.
Okay.
Thank you all.
Thank you all.
Thank you all.
Thank
Take
you,
care.
ma'am, for this wonderful session.
You're welcome.
Thank you.
And we will be having more webinars, and the announcements will be made on the website of FABA US chapter, as well as through our WhatsApp group.
Okay.
sir.
Bye-bye.
Sir,
Okay, bye-bye.
Thank
ma'am,
you.
please
Thanks a
recommend us
lot,
about
ma'am.
the internships also or the programs
Sure,
also.
definitely. Stay in touch and connect with us on the website. There's an email ID. So you can write to us at info@fabausa.org.
Thank you so much.
You're welcome. Thank you all.
Hello, ma'am.
Thank you. Bye-bye. Good night.
Yes,
Ma'am, is there any internship opportunity also?
we'll let you know if we plan something, end of the year or early next year, we'll let you know.
Okay, ma'am. Sure, ma'am. Thanks a lot.
Sounds good. Thank you all.